Okay, today we're going to talk about active transport. Before we go too far into active transport, let's review a little bit. So I want you to answer the following 3 questions. You can pause the video after you hear them. 1, what is a plasma membrane made of? What are the parts that make it up? 2, describe the difference between simple diffusion and facilitated diffusion. And 3, what are the 4 biomolecules that we find within the cell? You can pause the video right now and go through those. Okay, so active transport, we really need to focus on the plasma membrane. The plasma membrane, remember, is made up of phospholipids. A phospholipid has a polar head and a non-polar tail. Really important to remember this because we have to have, in active transport, proteins because we're going against the concentration gradient. Now in simple diffusion, we only have small nonpolar molecules going through the cell membrane or the plasma membrane freely because, well, they don't repel the nonpolar insides. And then in facilitated diffusion, you have to have a carrier protein which allows for the movement of particles into and out of the cell. Remember, there are 2 types of proteins used in facilitated diffusion. One is a channel protein, which is pretty much a pore that's open all the time, like in osmosis, where water can move into and out of the cell freely. And 2, you are going to have a carrier protein. Now, a carrier protein has binding sites for those molecules to fit into, so only certain molecules will fit into the binding sites and will move through there. And then we have active transport. Active transport is a movement of materials across the plasma membrane using ATP. Now remember, we move from a low concentration to a high concentration in active transport. So it's like reverse of passive transport. Also, it's kind of like facilitated diffusion in one aspect, and that's with the pumps, because they are going to have these binding sites like we had in our carrier proteins. However, these ones have to use energy and go in reverse. So the most common one is the sodium-potassium pump. We find this in all of our muscle cells. So the sodium-potassium pump is actually going to move sodium out of the cell, and we move 3 sodium ions, which are the red ions here. They move 3 sodium ions out of the cell and 2 potassium ions into the cell. Now, this is kind of cool because it allows for a muscle contraction and relaxation. Without these carrier proteins in our cell, we couldn't have a muscle contraction and relaxation, and actually our muscles would clamp up and stay there. We wouldn't be able to move at all. The other type is vesicles. In a vesicle, we move larger things like proteins, polysaccharides, aka sugars, and other large molecules out of our cell. And we do this in 2 processes. One is called endocytosis, the other is called exocytosis. In endocytosis, we move large molecules into the cell, exos moving them out of the cell. Now, endocytosis is moved in a couple ways. We have pinocytosis, which are very small molecules coming into the cell, and we have phagocytosis, which are large molecules coming into the cell. They'll actually grab on kind of like Pac-Man, engulfing that and bringing that into the cell. Now, they'll move them to places where they need to be digested or used, or they'll actually work as a lysosome itself and start breaking that down so that it can be absorbed by the cell. And exocytosis is just a reverse. This little bubble here will come attached to the cell membrane and then will open up, spitting everything out of the cell. It's getting rid of the waste products for the cell. Okay? You do have an assignment that you're going to be working on. There is an additional video that I'd like you to watch. Have a great day, guys.